Simultaneously, if you go and visit the Sanger Institute website, there is a program called Artemis, which we use routinely in our laboratory for genome comparisons in bacteria. So this all just summed up, and then I came to this. Probably what we want to discuss today is One Health concept. As already we are talking about that nothing in this nature works in isolation. We all are interdependent. Interdependence, when we talk about a concept of One Health is coming, this One Health concept says that the health of animals, humans, environment, this is all interconnected. So this is the One Health concept, and probably that's why I congratulate the organizers for taking up this theme, where we are basically talking about One Health concept. And for that reason, probably I will shift towards from the science to coming to the term environment. And sir, this environment, if you come into the Hindi, it's called Paryavaran. And I always find a better definition of environment with this term Paryavaran. What is Paryavaran? When we talk about Paryavaran and we see this word, probably the ethos of One Health is indigenous in the etiology of this term Paryavaran. Paryavaran, वो आवरण जो कि हमसे परे है, लेकिन वो हमारा ही है. That is part of us. That is the cover. That is the avaran which it's a part of our avaran beyond us. Beyond us, whatever it is Paryavaran. So in that case, probably we already have this idea of One Health in our Paryavaran or the environment. So environment, human health, animal health, all interdependent here. Then I gradually move towards to build up this story where I want to talk more and more about. That is, we already heard about fossil fuels, the use of fossil fuels, our dependency on the fossil fuels. In fact, nowadays, if you're interested in news channels, of course, nowadays it is more like the elections have been declared, so you will not find this. But this environment activism is a kind of thing which we find in our magazines and newspapers quite frequently, and you will see that stop using fossil fuels and don't use fossil fuels. They paint the orange color on, they block the road and kind of that. This kind of activism is very common in European countries, of course. But whether this is justified or not, I am not going to into this debate. But as a scientist, we always look towards solution. Whenever we come to see a problem, our dependency on fossil fuels, we always look towards some solution. Now, fossil fuels right now it is the main energy driving force on the globe. But the use of fossil fuel in itself is creating a lot of problems for the health, for the environment, and of course for the health. How it is interconnected, I will try to connect these stories together, and probably it will catch your attention also. So right now, fossil fuel is produced approximately 89.9 million barrels, but this is a limited resource. So this is the production all over the world, and this production and its use in itself not only create problems on the environment, but towards the human health also. So all of these things are interconnected. So according to this Intergovernmental Panel of Climate Change, the oil and gas industry is responsible for approximately 37% of global carbon dioxide emission. We are talking about climate change, and from this CO2 emission, because of fossil fuel combustion, has developed this need that already we heard, that we need fossil fuel to replace fossil fuels with the biofuels. The market of biofuels should come up. But fossil fuels, the problem which the fossil fuel use is creating, we need to address that also. How to remediate that, or how to take away that from the environment, how this problem should be, and it should be done in sustainable way. So this is one of our review paper where we have shown the amount of problem, the amount of this polyaromatic hydrocarbon and crude oil contamination all over the globe. And this is from the different time period, and the size of dot speaks about the amount of contamination. And as it is as you can see from this map, that this is not a local problem, it is a global problem which comes with the use of fossil fuels, and the problem persists and keep on increasing with time. So we have to see this challenge, but how to see this challenge is also a challenge. See, the use of fossil fuels, the production of fossil fuels also brings some undesirable events, like in Assam. I come from Northeast India, and the Northeast India is blessed with the resources, natural resources, and such one of the natural resources, crude oil production. But the crude oil production also brings such kind of disasters. There was Baghjan gas leak. We lost human lives in this accident, and not only that, there was acres, hectares of land, agricultural lands, which became unusable for agriculture. The biodiversity was lost, and it is lost for quite a long period now. How to reclaim these lands? It's a challenge basically. So, sir, you have already discussed a lot about circular economy, so I will better skip that, because we have already discussed in the morning session. We have already discussed this circular economy, but we can put the concept of circular economy for the bioremediation purpose of fossil fuels. How we can do that? That is where we come into the picture, and we are trying to develop some strategy using sustainable principles which is already available in the nature, and we are trying to resolve this problem using this. So I just skipped this. This is the bioeconomy of sustainable bioremediation approach. Now, bioremediation, as we think, is something which is nature-friendly thing, and it should be done out of goodwill or something like that. No, it is not like that. Bioremediation itself is something which the industry also sees from the perspective of economy, or from the perspective of currency generation. Say, if you look at the global stage, this is the picture which depicts. This is still to get published. It is in communication right now. So this is as per the European Union. It estimated cost for the soil restoration is $38 billion, and this is a very huge amount. And the European Union spent this amount to reclaim the soils, because we cannot. There is no alternative of soils. We need soil for everything. In fact, the target of 2050, where we will have to feed 50 billion of people on this planet, that depends on soil. So we have to reclaim the soil. The top 5 investors contributing to the world in bioremediation technology include US, of course, 61.85 in terms of patent filed. And if you see this picture, there are dots. These dots, basically, this gives high bioremediation market revenue where these countries are generating. Somewhere India is also there, although major part comes from this part of the world. So bioremediation is not only something which should be done out of goodwill, but it also has important role in the bioeconomy. So this is the picture which we commonly see in Assam. If you go towards upper Assam side, there are a lot of rigs, there are a lot of this oil excavation processes which goes on. So this kind of contamination takes place. You can see the crude oil which goes into the agricultural fields like this. And what are the effects of these crude oil going into the field? Now we are losing agricultural soils. Now these lands are unfit to grow food, unfit for agriculture. The crude oil in itself is a complex mixture of hydrocarbons, and these hydrocarbons, they are linear hydrocarbons. Linear hydrocarbons, they get attached to the soil, and it becomes the water unavailable kind of environment. So the plants cannot grow there. Microorganisms the agriculturally important microorganisms, they cannot thrive there. And along with that, it contains polyromatic hydrocarbons. Now polyromatic hydrocarbons, they are, although very lesser in amount comparatively, but they are carcinogens. They are cancer causing, and they are difficult to degrade. And high molecular weight pH, they are xenobiotic compounds. So this is all undesirable. We don't desire these things in the environment. So what we did, we did metagenomic analysis of some of the contaminated environments, and this is the diversity of organisms in these contaminated environment, and you will find although the contamination level, it was different in different area, the diversity also, it was different as anticipated. But the interesting part, which is significant for this conference, that I want to highlight. Okay, only 3% of the metagenome functional part was found to be able to degrade these hydrocarbons. That means we need something from outside. We need to have some intervention from outside to get rid of these hydrocarbons and reclaim these soils for agriculture purposes. And this is what interesting thing we found, that if you compare this contaminated environment with the normal agricultural fields, these fields were at these contaminated grounds were acting as hotspots for the development of ARGs, antibiotic resistance. So that's why I brought this story for this conference when I saw the Artemis antibiotic resistance, environment and industrial solution. So I thought I should elaborate this thing. And here you will find, so these were the microbial, these are the metagenomic analysis, culturable as well as non-culturable organisms, which were found to having contributing towards antibiotic resistance in the contaminated aromatic hydrocarbon as well as crude oil, this linear hydrocarbon contaminated land. They were contributing for antibiotic resistance. These are, I'm sorry for the lower, smaller fonts, but these are some of the antibiotic resistance genes, their abundances, and these are the organisms from, these are the different areas in which these were coming, and you can see that in the control land it was lesser, and all the contaminated lands they were having more ARGs. And if this picture, again, if you compare these two pictures, these are the colored dot you see is basically the microbial populations having crude oil degrading abilities, those gene functions. The dark dot you see is basically ARGs, antibiotic resistance genes. These are the contaminated lands and this is the control. And you can see the difference. That means these crude oil contaminated land and these bubbles you see is basically the taxonomic groups. So different taxonomic, in each of the taxonomic group you will find that these dark dots are more. What does it mean? It means that the antibiotic resistance genes, they are, no, imagine whenever we talk about antibiotic resistance, we always think that we should complete our antibiotic doses, we should follow the doctors. It is always the antibiotic uses that causes antibiotic resistance. But what if we look at the bigger picture, the One Health concept, where the environment is not healthy, then the antibiotic resistance are developing into the environment itself. So this is what we found, that the crude oil contamination and the polyaromatic hydrocarbon contaminations, they are responsible for the emergence of antibiotic resistance into the contaminated environment. So there we can found so the findings we could publish in environmental research and the science of the total environment, both are 10+. So then we thought that why is it happening? Although we work on the degradation, then we thought that what is the reason that crude oil and antibiotic resistance, they have, what is the commonality, common factors which is because of which this antibiotic resistance is there, and what might be the reasons? And then we came to certain hypotheses, and we came up with this. We found that the class 1 integrons, whomsoever who work with the antibiotic resistance, they know that the class 1 integrons, they are very significant point for the cross dissemination of antibiotic resistance, and there the efflux pumps, they play important role. Now the polyaromatic hydrocarbons as well as antibiotics, they overexpress this efflux pump genes, and because of that, this might be the one of the reasons. So we published this one hypothesis, and where the co-selection, we describe the co-selection process in the environment might be responsible for the development of antibiotic resistance in the contaminated environment. And the second one was in ESPR, where we have described how the mutation in the efflux pumps genes could be responsible for this phenomena. But again, this was very interesting. Now we are taking up in this lab, in our lab, and we are trying to prove this. So that work is presented. Maybe when next time when I come, I will come with experimental evidence. So now I come to the solution side, what we work, and that has to be sustainable. So what we found that usually what is done whenever we talk about bioremediation, we talk about bio-augmentation, where you have some organisms, some microorganisms which have good ability to degrade some contaminant, and you go and put that into the soil or into any of the contaminated environment. Now what happens that when you put these organisms into an artificial environment which is already contaminated, that contaminated environment always contains its own indigenous microflora. And indigenous microflora poses a competition to these augmented organisms. So because of that, what happens sometimes you might have heard during conferences and so that the formulation, it works good in the lab, but it doesn't work well in the field. What is the reason? The reason might be that we are talking about an individual to get fit into a community which is already well versed and very comfortably, stably present there. So we have to inoculate these organisms with a strategy where this community accepts it in a way that the contaminated environment accepts this applied organisms, and that's why we tried to go for rhizoremediation. And what is rhizoremediation? Again, we are using the concept which is already available in the nature. Here we are using plant roots and the bacteria which grows on the roots as the injection system to go in the soil in the contaminated environment. Now the root, it goes into the soil. Wherever the root grows, the bacteria grows along with it. We call it rhizobacteria. Its natural habitat is the root surface. The root produces the root exudates, the nutrient for the bacteria. So bacteria takes that nutrient, goes into the soil along with the roots, and wherever it finds contaminant in the vicinity of roots, it degrades it. So it catabolizes the contaminant. So here it's a win-win situation because the plant is getting benefit from this bacteria, its growth is improved. These are plant growth promoting bacteria, and the bacteria also reclaiming the soil. The contaminant is being removed. And also the indigenous organisms, those organisms are supposed to welcome this because it is coming through plant roots, so they are also getting the nutrient. So while translating this natural symbiotic phenomena into the environmental application, we find a sustainable solution to remove the contaminant from the degraded lands, from the contaminated lands, and that is what we call rhizoremediation. So I have been working with this rhizoremediation for approximately a couple of decades, so I cannot see all the data here, but some data, some stories I have brought for you. So we screen for phosphate solubilization, siderophore production, SSCD managed activity, nitrogen fixation. All of these are plant growth promoting activities which we see in these organisms, that these organisms can degrade the crude oil and polyaromatic hydrocarbons, and along with that it can also promote the growth of plants. So that is what we check, and then we select these organisms. Now see, these organisms, they have multiple attributes now. They can colonize plant roots, they can degrade the hydrocarbons, and simultaneously they can also promote the growth of plant. So these organisms we select out of these organisms. So this is some of the data. We select some of the organisms, then if you see the genome of this, one of such organism is Pseudomonas fragile DVC, and if you see the genome of it, you will find these attributes in the genome of this organism. So this is green for pH degradation, then in rhizosphere environment it has to survive, it has genes for that, biofilm production, nitrogen fixation, phosphate solubilization, all its genome is full with all those beneficial attributes. We need such organisms. So little bit of molecular work where we find, now sometimes what happens, the challenge, we feel that sometimes these organisms are good for soil, living in soil, but their efficiency cannot be improved in terms of degradation, catabolic activity. So we have thought to improve their catabolic activity to can increase through some genetic manipulations, like BEN-D is one of such gene, which is a transcriptional regulator. So we have targeted this BEN-D gene, a transcriptional regulator. Little bit of science there. So for microbiology students, some experiments we have done with this BEN-D, and we have tried to characterize this XYLS type of transcriptional regulator, the BEN-D product, and it's a promoter binding protein basically, and we have tried to improve its binding capacity so that the degradation ability of these organisms should get improved. But one challenge what we face is that the genetically manipulated organisms cannot go into the environment as per the regulatory affairs. So that's why this work is of course for theoretical benefit only, because you cannot take this genetically modified organism into. But of course, this is one interesting place. Someday, if we can get permission, and if the biosafety committees allow us to do that, so these organisms can perform better into the environment. So this is one of the paper out of it. Another interesting thing, sometimes this is for microbiologists only. Many times while working with this, such degraders, we found there is some organisms which are called to be pathogens, human pathogens. We isolate, and they give very good activity in fields, say like Klebsiella, Burkholderia. Burkholderia is an offshoot of Pseudomonas taxonomically. So one of the organism was AWD5, which we were working, and it was Klebsiella pneumoniae. And Klebsiella pneumoniae, it was degrading. We published in this data, but it was very difficult to get it published because every reviewer was saying, okay, fine, this is a pathogen, you cannot use it. So we took it as a challenge, and we did animal model pathogenicity testing, and this we did complete genome of it. And what we found that this organism is totally sane. It does not cause any infection. This is the mice model experimentation, where the control and the infected mice was alike. Its histology was alike, its organ conditions were alike, everything was same. So it was a non-pathogenic Klebsiella pneumoniae. So we published it because somebody said to us that this organism you should omit from your work. We did, because again the regulatory affairs will not allow us, but of course we publish it from the scientific perspective, because microbiologists, we know that we name the organisms on the basis of 16S sequence, but it is the complete genome which is working there. So this is a different perspective from the side of a microbiologist. So I thought that I should bring it to the young generation. Maybe somewhere we have to find a solution to this, where the yardstick of 16S does not define the functional attribute for organisms, or guilty until proved. So yes, another organisms, alkali genes, faecalis, similar data. Then the pathways for degradation of polymetallic hydrocarbons, and similar kind of work where we describe what pathways they are taking basically, because these aromatic hydrocarbons, they are carcinogenic, and their intermediates are also very toxic. So that's why we want the complete removal of these polymetallic hydrocarbons as well as their intermediates into carbon dioxide, which happens by the bacteria by taking these intermediates into TCA. So that is what we do with this is another. Now this crude oil contamination also brings challenges where it's not the environment is not only contaminated with crude oil or its complex hydrocarbons, but it also comes with the heavy metals, and other pollutants are also there in the same environment. Pollutants, the contaminants are never comes in a pure form in the actual contaminated site. It comes with a lot of other contaminants as well. So that's why we try to get organisms which also have heavy metal resistance, and that is what we get, like Serratia marcescens. And yeah, this picture must be little interesting for this conference. This is a genome-wise comparison between Serratia. One is from the environmental isolate, one is rhizospheric isolate, but these blue-red lines is of course to show the homology between the complete genomes, but this has been produced by the software known as Artemis. So this is Artemis product basically. So this is your conference theme, Artemis, and this is Artemis product of genome comparisons for Serratia species. So here again, this we can see this mainly as a directive. We reported for the first time that this plant can be used for bioremediation of crude oil, for polymetallic hydrocarbons, along with the resistance against cadmium and other heavy metals. So this is benzopyrene from Acinetobacter. You can see the diagrams here, the biofilm formation, and which is also one of the important criteria for crude oil degradation is its bioavailability. Hydrocarbons are not bioavailable to the organism. Even if the degrader is there into the soil, it is not bioavailable because it is very hydrophobic. It makes the soil very hydrophobic. So one such molecule, a set of molecules which is important for to increase their bioavailability is biosurfactants. Now biosurfactant production in itself is a different industry altogether. Biosurfactant, it finds its application in several other industries as well. So here also we tried to have a biosurfactant producing microorganisms, rhizobacteria, which promotes the growth of plant, degrade aromatic hydrocarbon and also produce biosurfactant. So we were successful and to get such organisms and their genomic and genomic attributes we reported in genomics. So now, while I present this work into different conferences, one of the questions which used to come to us that how do you select these plants basically, because here the plants are also important because it's a plant microbe interaction, and to sustain that contaminated environment, it is not only the organisms which is important, it is also the plant which should be resistant and it should be able to survive there. And as a matter of fact, it is the plant who is the weakest link in this when it goes to grow into the contaminated environment, because plants are more vulnerable to the contaminated environment than bacteria, and that is why this is one of the challenges. So what we found that while using this rhizoremediation strategy, our bacteria was not only promoting the growth of plant but making it more stress resistant, or say giving that it's more strength to survive in this stressful condition which is imposed by the contaminant, that is the hydrocarbons. And that is what we did by this glutathione and glutathione transferase mechanisms. Glutathione metabolism in plants are known to counter the stress which is imposed by contaminant. These glutathione molecule, it binds with the toxic compounds and it takes it away from the system. So that is what we analyze in the plant. And in plant roots, which is in direct connection with the contaminated environment, we found that the glutathione mediated metabolism, it was taking good care of the plant, the stress level was taken care. So that means our bacteria was not only degrading the contaminant but also making the plants more favorable to grow under this stressful environment. So this was an interesting find, and we could report it for the first time with using this Jatropha as the model plant and Pseudomonas aeruginosa. So Jatropha is also one biofuel plant. So I will talk about a little bit of circular economy, how we are connecting it with our rhizoremediation. Then another question which somebody may ask, when the plant roots are providing so much of nutrition to the bacteria, why bacteria will utilize the contaminant as for its catabolism or for its metabolism? Of course, when you are getting one source of food in your plate, what is the need to degrade the difficult one? So this is one question which we always face as a challenge. So we designed some experiment for that. We did some analysis. We did some RT-PCR analysis there and some transcriptome analysis there, and we found that Pseudomonas is something, some organism which is a very, very interesting organism. We already know it has a reverse CCR, reverse catabolite reparation. And here we could design this experiment where we provided synthetic rude exudates. We made it in our lab, and this organism was able to degrade hydrocarbon along with the rude exudate nutrient, and we checked it through the expression of catechol 1,2-dioxygenase and 2,3-dioxygenases. These are the two enzymes which are important for pH degradation. So this is the set of experiment which we put, and we gave benzopyrene along with amino acids and glucose, then organic acid, amino acid, all of the mixture here. And what we found, thankfully, that this organism, this is the experimental condition, this organism was able to degrade these compounds. The genes were expressed in spite of the presence of other carbon sources. Although there were some differences in presence of amino acid that we were unable to explain, but in the presence of other carbon sources, these genes were getting expressed almost in the similar level. So this makes us a little bit comfortable, and we were able to answer, no, there's no problem. If the root exudate is there and the contaminant is there, still we will be able to degrade the contaminant. So again, this is another, this is catechol 2,3-dioxygenase. The previous one was catechol oxygenase, and then we would we published this in ecotoxicological and environmental safety. Then right now, microbiology is going through transformation, as you see. From last couple of decades has been very interesting when this next generation sequencing has came into picture, and this has given us ability to study those organisms which we don't get on our regular culture plates, and we call it unculturable organisms, although it is a wrong denomination, because today or tomorrow if we culture it, it becomes culturable. We cannot culture it because we don't know the growth requirements of these organisms, and only, as a matter of fact, only 9 to 10% of organisms are culturable by this definition. Rest 90% of organisms we have not cultured yet. That means 90% of the potential of these organisms are still yet to come into our mainstream of microbiological applications. So because of that, this the same concepts apply in soil microbiomes also. Now the microbiome study is developing more for the human gut microbiome. You will get a lot of nature paper in gut microbiome area. But what about environment? What about agriculture? So it becomes our responsibility to see the microbiome studies into these areas as well. So there are some of the definitions for the students. What is core microbiome and satellite microbiome? So these are a little bit theoretical, so I just skip it. Or if you are interested, what is core microbiome? It's that part of microbiome which doesn't vary much among the individual despite of the environmental variations or other variations. Some part of the microbiome remain constant in an organism. Some of the part it remains dynamic. So which is constant part that we call core microbiome. And this is basically we try to maintain that core microbiome as an indicator of health. If the soil has that microbiome of a particular region, the core microbiome remains as such that we can say that it is a healthy soil. And whatever the diversion from the core microbiome, we call it dysbiosis. Dysbiosis for our gut microbiome also, dysbiosis. If the microbial community, if the health is not good, the gut microbiome will shift from, core microbiome will shift from core microbiome, it will be different basically. So the dysbiosis will occur. So this concept we try to apply for designing microbiomes for desired effect. This is called microbiome engineering. Microbiome engineering, where you can engineer of course we cannot do it directly because we cannot cultivate it, but we can do it indirectly. So again, this is an interesting field of research where you can design the microbiome as per the requirement, whatever the requirement you need. If it is for your health, you can design your microbiome through whatever food you are taking or for whatever things you are doing. If I take a simple example, there might be a situation where some problem comes to you and you can handle it with very good confidence. And in other situations, the similar or even lesser extent of that problems can cause an anxiety inside you. You know that anxiety is directly affected by the gut microbiome of yours. So if you maintain your gut microbiome, you can face your problems better. Similarly, if you maintains the microbiome of soils, your crop will be healthy. If you maintains the microbiome of an environment, it will remain contaminant free. So that's what is the basic principle which we try to translate here also in the soil. So what we proposed, that the rhizoremediation strategy which we are using can be, we published this hypothesis there, that this rhizoremediation strategy can be a tool for the microbiome engineering in the contaminated environment, and that is what we propose here.
in one of our paper. And later on, what we propose here, that the major part of microbiome is basically
it's basically governed by n number of factors.
But if we can maintain this small amount of microbiome through rhizo-remediation, through application of plant as well as associated microorganisms, it maintains a core group of microorganisms which is always healthy, and this will facilitate improvement of the quality of that microbiome, and that is how we can engineer the microbiome in the soil so that it remains healthy and it has a self reclaiming property attribute because of this. So that is how we did, and then the hypothesis as well was published, and after that it was our responsibility to support this hypothesis. So we designed experiment for that, and we designed a couple of experiments with plants and bacteria, and then we with these are the group of organisms. So we then analyzed the microbiome after rhizo-remediation, and we found that this hypothesis works very well. The rhizo-remediation can be an instrument for the microbiome engineering, where we correlated this relative abundance of certain organisms. You can see here, and some of the organisms, say X is there, then Y will be there. If X is not there, Z will not be there. So similar kind of observation we made among the different microbial genera and also the functional genes as well. So this is how we found that this can be done. We can modify the microbiome by using plant microbe interaction, and this we were able to publish in two different papers, into two different set of experiments. Applied soil ecology, and the second one was with benzopyrene with Melia. It was published in Chemosphere. So that's how we were able to support that. Okay, fine, rhizo-remediation can be a tool for microbiome engineering, and this was the first report where the microbiome engineering was used with the rhizo-remediation aspects of the crude oil contamination. Now, these are all theoretical things, but whether or the say lab-oriented things, whether this thing work in the field as well. So for that, we went into the field, we took up the trials, we went into the field, and we went into crude oil contaminated areas. We took our formulations, we took our plants, and we applied that here. These are different treatments into the soil, and this was one with the other director indica, and here are some of the treatment and the plant enzymes, and we could establish that the plant growth was improved. The plant without bacteria in fact it was very poor growth. In itself, the plant were unable to grow in that contaminated environment, but when our consortium was given, those were growing very well. And this is we did the microbiome analysis again into the different contaminated environment after the treatment. This is something interesting. Okay, fine. This is you can see the plant, how the plant they suffer in such contaminated environment. Just look at this scanning electron microscopy of the sections of the plant. You will see this is the control. Do you see the difference between here, and here? These are the tissues which are facing the contaminated environment, and these are the tissues, and this is the control. So that is why I said the plants, they are more vulnerable. But when these are the depositions of PEH you can see inside the plant tissue. The plant uptake. Plant do not degrade, they uptake. Now, when we realize this, that the plant is uptaking the contaminants and accumulating in the tissue, and now if the plant is grazed upon or goes into for human or animal consumption, it's a problem again. So a problem should not be a solution of another problem, isn't it? A problem should come with a solution, not another problem. So what to do with this plant? Then we thought about going about the circular economy concept, and we prepared biochar. So you were asking about biochar. So we prepared biochar with these plants. We also tried to develop this bioethanol production using this plant material. And in itself, here we talk about climate change, carbon sequestration. Here in itself, the basic concept of rhizo-remediation, here the plants are growing, the contaminants then it is being degraded into carbon dioxide. The carbon dioxide, instead of going into the environment, will be trapped by the plants, and that we have experimentally established. And this, so if you compare with the normal bioaugmentation, here again we are contributing towards climate change, because the carbon sequestration is automatically there because of the application of plants. And then this plant, if we could take it for the biochar production or for bioethanol production, this completes the cycle. Now, coming to the final part, that we did after application of, because I initiated with that antibiotic resistome inside the contaminated environment. I started the story from there. Then what happened when we applied this rhizoremediation strategy in those contaminated fields? What we found that, okay, this is the after the treatment, this is the correlation of bacteria with respective degrading genes. What genes were coming from which bacteria? I will come to this slide later. I just move ahead. This is the taxonomic data, variations. This is the data which I wanted to show. This is again yet to be published, but if you see, this is a bubble diagram, and these are the antibiotic resistance genes. And if you see this antibiotic, the first one is untreated. That means we have not treated this. Just see the bubble diagram. And these are treatments. Here only plants, here with consortium. You can see the bubbles have gone smaller. The size of bubbles shows the relative abundance of ARGs into the environment. So rhizoremediation, contaminated environments in the field was giving rise to antibiotic resistance. But as we put rhizoremediation strategy there, successfully remediated the soil, along with that, this is additional benefit that the ARGs were decreased. So we find a solution from the environmental concepts. One health. One health. Now, this antibiotic resistance which is generating in the contaminated environment will reach us someday somehow, isn't it? Now, but if we treat that emergence right at the source, then this is not going to come to us. And that is how the concept of One Health work. So that is what we observed here, yet to be published. So this is the patent which came out of this work, and these are the contaminated sites. This is which we put up a pilot scale study with in collaboration with ONGC, and there we remediated their soil. They were very happy. The black color soil was totally gone. It was we used populus and this populus plant for that, and they were very happy. The ONGC people were very happy with our strategy. In fact, there is one strategy also, oil zipper. If you know, there is one strategy of formulation from TERI, which is being used by this oil industry for bioremediation, but they use bioaugmentation strategy. For the first time we introduced the rhizoremediation strategy, but we are looking for its commercialization right now, discussing these things with the industry, but it was very successful at ONGC site. And along with that, there is one component of crude oil which nobody talks very much, because it is one of the toughest component to degrade. And this is this one, asphaltene. Asphaltene is also part of crude oil, and asphaltene is very difficult to degrade. If you see the structure, it is very complex structure, and for the first time we reported that the asphaltene degradation from rhizospheric organisms for rhizoremediation. So we were able to deal with this hardest part of contamination of crude oil as well. This was published in Bioresource Technology. This is the word I was talking on it. Then now I come to the slide which I skipped, and I don't want to skip it because it is very challenging things which get answers, because one may ask that while choosing, okay, somebody is knocking my door, I don't know who. Somebody asks, some may ask to us, okay, fine, bioaugmentation, I take bacteria into the soil, you saying indigenous organism replace it. What about your organism, whether it is getting replaced or not? indigenous organisms may replace the same organism which I am putting up with the plants as well, isn't it? It is I am hypothesizing that the roots will take it along. We need experimental evidence. What to do that? So for that, we tagged our bacteria with GFP. You can see this green signal coming. GFP is a gene which comes from the jellyfish basically. So none other bacteria will have it. You will not get it background noise for GFP if in any other bacteria. So we tagged it with the GFP protein, and our bacteria starts glowing under UV. So we colonize with this is the root part, and you can see the growth of these bacteria around the root hairs. So this bacteria was growing very well around the root, and that is how we could be in the presence of indigenous organisms, which we showed by metagenomic data. So again, we were able to answer this question that no problem. Our bacteria is, this is called rhizospheric competence. It has to be competent in the rhizosphere. So that is how we were able to complete our story with all justifications. Okay. So this is one of the patent, and this is the work which has gone on the ground. Take home message: rhizoremediation approaches may offer efficiently reliable advantages as compared to other bioremediation techniques. I am not saying other bioremediation techniques are not good and rhizoremediation is good. I am not saying that certainly, but of course rhizoremediation may be used as preferred, or may be amalgamated, or may be used along with the other strategies to get a healthy environment. And because it relies on the natural processes, and we are taking the natural concepts, we are deriving the technology, the strategy which is already available in the nature in the form of symbiosis. We are just amplifying that effect by taking a microbial consortium, a formulation along with a suitable plant, and taking to the contaminated land. So these are other papers from the same work, and acknowledgement to Ministry of Environment and of course DBT, which has been very kind. This is my team. All credits to them basically. I am just telling the stories which they have generated. And Ritu is right now in Denmark, postdoc. Anand is a scientist. He is faculty in University of Rajasthan. His wife Malvika also is faculty in Rajasthan, Central University of Rajasthan, and others are also working in different educational institute. So this is all credits to them. I never fail to mention their credit because it is them basically who has generated such good data for this. So thank you. This is my university, a beautiful campus. Whenever you find time in that part of, just visit. If you have planned something to visit Northeast, come to Shillong. Is a beautiful place. You can plan and come there, Shillong. Whenever you come Shillong, I am just 6 hours distance to Shillong. Just visit my place. Thank you so much. Thank you.
Thank you very much, sir. It was indeed a spectacular talk. We would like to extend our thankfulness with a memento. And for this, I would like to invite Dean Research and Innovation, Professor Anand Tiwari, sir, to extend our thankfulness with a memento to very charismatic Professor Piyush Pandey. A big round of applause. Thank you, sir. A small announcement. We have a QR scanner here. If you have any questions, you can post there. So next, moving ahead, we have our second invited talk, and we are happy to welcome Dr. Dwapan Goswami from Gujarat University. Dr. Goswami is an luminary in the field of biotechnology, showcasing scholarly excellence grounded in a robust academic background. With a PhD from Chalodar University of Science and Technology and a commendable 36th rank in CSIR NET Life Sciences in India, he stands as a paragon of academic achievement. Presently, he is serving as an assistant professor at Gujarat University in the Department of Microbiology and Biotechnology. Dr. Goswami leverages his expertise to foster innovation and disseminate knowledge. His scholarly impact is valuable through a stellar citation record boasting 2,557 citations alongside an impressive H index of 21 and I-10 index of 40. Dr. Goswami's prolific contribution to scientific literature encompasses 57 articles, 14 book chapters, and numerous papers in conference proceedings. He demonstrates unwavering commitment to research leadership, notably as the principal investigator for prestigious various projects like GSBTM funded network program on antimicrobial resistance and an UGC funded study on trichodoma. Beyond research, Dr. Goswami plays a pivotal role in shaping scientific discourse as an esteemed editorial board member of Scientific Reports. Additionally, his educational outreach extends to his YouTube channel Learn at Ease, where he simplifies bioinformatic concepts, amplifying academic research and accessibility. Dr. Goswami's expertise in bioinformatics is instrumental in drug delivery development and addressing antimicrobial resistance through genomic analysis. Recognized as one of the top 2% of global researchers by Elsewhere, his curriculum vitae underscores a profound dedication to advancing microbiology, biotechnology, and bioinformatics for the betterment of human health and sustainable practices. Let me take this opportunity to invite very energetic Dr. Goswami to share his expertise. Over to you.
Thank you. So first of all, am I audible? Yes. So I have 2 tasks to be done today. First is to complete my talk before the lunch break. I absolutely understand that you are sitting over here since a pretty long time. And the second thing that I want to do is to motivate you, because we are living in a realm where fear is one of the most important thing that is being, which is being eating us up. So today I will be talking about antimicrobial resistance. But my goal is not to just talk about antimicrobial resistance. My goal for today is to make all of you aware that how you want your future to look like. How you want to see your future. Do you want to be under this fear for the next 50 or 60 years of your life, or you want to actually be a good researcher in future? I want to answer, or I want you to answer yourself, retrospecting what your thoughts are. So before I begin my talk, I will split this talk into 2 major sections. The first part is that we will be understanding about what is antimicrobial resistance and why we are so much worried about this kind of resistance. And as also sir mentioned, we are also talking about ARGs, that are antimicrobial resistant genes. So if a drug is there, you are taking the drug, the bacteria are getting killed, then how the hell these genes are coming into the picture? What are the functions of these genes which are very important, which is causing these microbes to become resistant to something? Now, before I dive into the topics of antimicrobial resistance, I first of all want to tell you the terminology which is even bigger than antimicrobial resistance, that is called as, in general, drug resistance. So now what is basically a drug resistance? So when we are talking about the clinical terminologies here and there, so basically drug resistance is something where any kind of drug is not functional. Even if you talk about, say, cancer. So cancer drugs, the drugs which are basically designed to cure cancer, if it stops working, it is called as drug resistance. If a drug is designed to cure certain kind of a disease, if it stops working, it is basically called as drug resistance. Now this is a big domain of drug resistance, and within this domain, a smaller domain that falls in is called as antimicrobial resistance or antimicrobial drug resistance. So the bigger picture is drug resistance, smaller picture is antimicrobial drug resistance, and within this antimicrobial resistance, we are talking about the drugs which are applicable for bacteria, for fungus, for viruses. But the problem is that most of the diseases that are being caused to humans are bacterial. This phenomenon is seen in bacteria. This phenomenon is also seen in fungus. But looking at a bigger picture, we are more fearful towards bacteria. Talking about fungus, in 2020, just after the completion of corona, there were a lot of incidences about mucormycosis. And as soon as we came across those kind of scenarios where mucormycosis was there, WHO has given a verdict that we also need to look into the antimicrobial resistance in the domain of fungus. Why? There are a lot of infections that have been caused by yeast. There are a lot of fungus infections that are also being caused by molds. So today I will not be focusing on yeast, I will not be focusing on molds, but I will be specifically focusing on the bacteria. Why? Because this is the major fear domain that we are diving into. Most of the researchers are worried about this problem, and this is a serious problem. I will come into it. So, this is a very important slide because this will build up on the things that I will be talking in the next part of the session. So, basically we have major 5 classes of antibiotics. The first class of antibiotic that we talk about this are the antibiotics that will stop the synthesis of the bacterial cell wall. So if you basically block the cell wall synthesis, what will happen? The bacteria cannot multiply, or even if it multiplies, the bacteria without a cell wall, it is very difficult for that cell to survive within the human body. So it will basically die. The next part is that it will basically target this central dogma which is there inside the human body, where replication, transcription and translation. So these are the 3 major mechanisms which lay down the central dogma. So if you block one of these, then bacteria cannot do its function. It cannot synthesize protein, it cannot replicate. Okay? And the third one, and the most important one, is certain enzymes which are very crucial for the survival of the bacteria. If you block those enzymes, what will happen? The bacteria cannot survive. So certain antibiotics will focus on the certain pathways of the bacterial system, and therefore the bacteria will stop multiplying. So this is how basically the antibiotics were developed. We are being using these antibiotics. Now comes is the picture, inside this picture is the antimicrobial drug resistance. So these are the main domains. So basically what we can do, antibiotics has to go inside the cell, or it has to interact with the cell. So if antibiotic does not enter into the cell, then that antibiotic will not work. So for that, sir has also mentioned efflux pump. The antibiotic will go inside the cell, but bacteria will throw them outside. Now for each of the efflux pump, what is efflux pump? Basically it is a protein or a transmembrane protein. A protein comes from a gene. If this gene is present, the protein will be present. Protein will be present, then there will be the transmembrane protein which will throw out the antibiotic. So for each function there is a gene which is responsible, which is basically disallowing the disallowing the antibiotic to function in a particular way. So as we can see over here, what we can see is that antibiotic, first of all, the entry has been blocked, means this is the antibiotic. So bacteria is evolving at a much faster rate than the speed at which we are creating new antibiotics. So what will so this is one of the mechanisms. Second mechanism is the alteration of the target molecule. So assume that the antibiotic is basically binding to the enzyme and blocking the enzyme, and I hope all of you know that enzyme inhibition is very crucial. You might have also studied competitive, non-competitive, uncompetitive kind of enzyme inhibition. So basically these antibiotics can also work as enzyme inhibitors. But what if there is a mutation itself in the enzyme or the protein to which it is binding, and that mutation can be directly correlated to the gene which is there. So mutation in the gene will cause the mutation in the protein, and ultimately that protein will not bind to the antibiotic to which it was designed for. Then certain bacteria will produce a unique kind of enzymes in such a way that it can basically eat up the antibiotic. So what you are feeding the poison to the bacteria, but bacteria has evolved to such a rate that now that poison is a food for a bacteria. Why? Because it has acquired certain genes, and those genes are making certain enzymes which are basically degrading the antibiotics. So what is antibiotics? Antibiotics are nothing but a carbon, nitrogen, sulfur. All these molecules, bring them together, make a compound chemical structure, and that is an antibiotic. So if bacteria acquire certain genes which will produce certain enzymes, and those enzymes will eat up the antibiotic, and now the antibiotic which were earlier a poison for a bacteria has become a food for a bacteria. And it is possible, if you see penicillin resistant antibiotics, you just take a penicillin, put it into a media, just water and antibiotic and agar, your bacteria will grow on it. So now once a poison, now a food. Okay. Then the next is efflux. So now let me just go about and just talk about the one single phenomenon. And now, when I say about replication, quinolones. Quinolones are one class of drugs which basically blocks the replication within the bacteria. So the DNA cannot divide. If the DNA cannot divide, the bacteria cannot divide. Now what happens is that this drug basically targets a gene which is called as topoisomerase. And the gene which is responsible for topoisomerase is known as a gyrase A gene. So gyrase A will produce a topoisomerase that is been basically targeted by this antibiotic. Now what has been seen is that when you keep on giving these quinolines to the bacteria, it will start producing mutations inside this gyrase A gene. And it has been observed that there are total 7 different mutations that takes place. 7 different mutations within the same gene caused by bacteria to overcome this antibiotic. So this is where the genes comes into the picture. So I can talk about this on a multiple levels. Talking about this beta lactamase, I just want to tell there is a gene which has been identified, or a enzyme that has been identified. It is called as beta lactamase New Delhi metalloprotein. It is a variant of penicillinase identified, or beta lactamase identified from the MRSA. MRSA is methicillin resistant Staphylococcus aureus. So MRSA from New Delhi. Now we don't know how many different kinds of enzymes like this are there, because we don't have the surveillance power. I will come to it later, don't worry. Now I also want to talk about one very important an example, like enzyme TETx. Now this is a gene, TETx gene. What does it do? It will produce an enzyme which is called as a TETx. So TETx gene will produce an enzyme which is called as TETx. Now this TETx gene will basically do an oxidation on the antibiotic. So basically there are genes which will produce certain enzymes which will do simple oxidation and reduction to the antibiotic, making the antibiotic ineffective. So this is how the genes comes into the picture, and this is how basically the genes work. Now this graph is very important. So now when we see, when the antibiotic dosages are very low, what is happening? The concentration of bacteria is very high. There is a point of crossover when you increase the concentration of antibiotic, the concentration of bacteria will fall down. But then if you keep on increasing the concentration of antibiotic, there are some survivors. And these survivors are those which have done some kind of mutation within themselves to survive. And you are all getting the infections of the bacteria which in some or the other way in past would have survived from some another patients. Okay, so and then comes the picture of unhealth. Why? Because we are taking antibiotics, we are throwing the antibiotics, it is going into the nature, bacteria are again surviving. So right now all the microbes which are there inside our body, which is there inside the soil, inside the river, some or the other point they have encountered the antibiotic and then they have survived it, and so therefore they are still alive. And that is how the things are moving. And we also know that these genes are basically not found in the genome. They are on the what is called as plasmids. And one bacteria can give plasmid to the another bacteria, and so the genes can be transferred. So this is the problem that has been seen, and that is the reason why there is a lot of issue about this antibiotic resistance. Let me come to the point. So now what is basically happening? We have identified certain, these are the crucial genes that I will be talking about. These are all the certain important genes that we know that suppose if you have a gene inside the genome of a bacteria which is called as TETx, definitely it will do some redox reaction on the tetracycline, and tetracycline will not work. So genes are there, do some activity on the antibiotic, antibiotic will not work. So this is the overall picture. And why we are talking about genes right now? Why do we need to talk about genes? Now let me make this thing very clear. We have a bacteria. Bacteria, you throw them an antibiotic. Bacteria is not been killed. And when the bacteria is not being killed, people are thinking, what the hell is happening? There has to be some genetic mechanism which is not allowing this bacteria to be killed. And then comes the power of the genomics into the picture. So people started doing the whole genome analysis. And then they identified, oh fine, this bacteria has acquired this number of genes. And so therefore, this bacteria is therefore not sensitive to this many number of antibiotics. Now this process takes a long time. And we do not see any possible solution to it. Because we cannot produce antibiotics at our will. This is a problem. This is a huge problem. Not said by me, said by WHO. Now, over the course of time, we have seen WGS, and this is going to be the center stone over the period of years as well. I will come to it. Now, just imagine, what is the cost of a nutrient broth 10 years back? Almost identical to what you have right now, or even expensive. What is the cost of whole genome of a bacteria today as compared to 10 years back? Which technology is getting cheaper? Genome sequences is getting cheaper. So now, imagine a scenario, what is basically happening? Today a patient goes with the lungs full, with a pneumonia, the patient is in the ICU, then a sputum sample is taken, it goes to the clinic, the practitioner or the clinician will basically try to isolate the bacteria from it. That bacteria is then tested by different antibiotics by the methods you all know, this diffusion or any kind of method that you can say. This process takes 7 to 8 days or even 4 days at the maximum. By that time, the patient is critical, he is under the ICU, doctor doesn't know which antibiotic to give. And by the time the results are out, the patient is dead. And there are thousands of examples like this. So what solutions we have? And my major motto is to give you what visions you need to have to overcome such problems. Let us go further. So WHO on AMR in the year 2015 global action plan on AMR was basically implemented that we should all do a surveillance. Now why the hell we should do the surveillance? The idea behind the surveillance is that what are the genes that are present in the genome of E. coli found in India compared to what is found in USA. Suppose if E. coli is having 15 different genes that are found for AMR in USA is known, but that same data we do not have for India. That is not it. That is not it. That is absolutely not it. We still don't know, are there any next level mutations within those genes or not. Now TETx gene, let me take an example of simple TETx gene. You have somehow created a drug for TETx. But what if before you synthesize the medicine, the mutations are already been introduced in that genome and 50% of bacteria are directly, even before the drug is into the market, is still resistant. We do not have a picture. We do not have a roadmap. We do not have a map where which mutation is there in which gene. I'm talking about the mutation, not the number of genes. We still don't have the information about the genes itself. If you take an example of E. coli, we don't know what is the pattern of gene panels which are there in India compared to Sri Lanka, compared to Africa, compared to Western countries. So we cannot simply take the medications which have been given in the Western countries, because those and the bacteria that you see, as sir mentioned, Klebsiella pneumoniae, the most feared, it is in the WHO critical pathogen list. One strain can also work as plant growth promoter. So we do not know what is the overall picture or the genetic landscape of those organisms which are causing infection in our country. And because of that, we are also working very hard. We are doing surveillances. Those organisms which are identified are sent for whole genome sequencing. It is a humongous task. But at the end of such projects, we will have some data to work upon. We do not have antibiotic policies. Why we do not have antibiotic policies? Because we don't know which kind of genotypes of those bacteria are still there in India. Not talking about India, what kind of genotypes are there in Gujarat or in Ahmedabad, we still don't know. We just know certain information that, okay, fine, carbapenemase resistance is found 40% in E. coli in the region of India. That's it. Even if you do a lot of hard work, this kind of data you can get. How can you cause each patient going into ICU with tuberculosis to undergo whole genome analysis of the bacteria from that sample and to have the clear picture? We don't have answer, but future is going to be bright, trust me. Yeah. So then there is something which is called as GLASS, Global Antimicrobial Resistance Surveillance System, and this is the exact reason why such programs were developed by WHO, by United Nations, for which India is also a part of it, where we are being guided that we should undergo as much as surveillance as possible to get as much as idea about the antibiotic resistance which is there inside your population where you are living. Again, of which 97 countries and 66 have reported the data for AMR. Right now India is also working on it. Now I will skip this part. So basically antibiotic resistance is broadly categorized into these categories: AMR, MDR, XDR and PDR. Stands for antimicrobial resistance, multi-drug resistance, extensively drug resistance and pan-drug resistance. So I will not go into the details, you can understand this later as well. And now this is something which is very important. So doing surveillance is very difficult. Should I do a surveillance on E. coli? Should I do surveillance on Klebsiella? So WHO has given guideline that if possible, you need to do surveillance on Acinetobacter, Pseudomonas aeruginosa, Enterobacter DSE. Now when we talk about Pseudomonas aeruginosa, it is one of the very crucial organism for xenobiotic degradation. So we are throwing them on one hand in the soil. We should know what we are throwing inside the soil. Then we have second priority, which is a high priority, and the medium priority. So these are the organisms which WHO said that as much as possible you need to do such surveillance on these organisms to get a clear-cut picture about it. Now some very important facts I want to tell about MRSA. It is observed that 29% MRSA prevalence was there in 2008. The number has gone up to 47 by 2014. And we are right now, this data is itself 10-year-old. What might be the scenario? C, very crucial part. Antibiotic ho, na ho, you have to stay healthy. You have to eat good food. You have to sleep well, keep your immunity high. Because you don't know, if you catch one of these, God saves you. Okay, now it is not so dangerous, but still I need to make you, yeah. So we have a national action plan which we have implemented in the year 2017 to 2021. Even Narendra Modi promoted it, that we should look into it. We should know, we should need to have a data of India. Folks, we are so many researchers across the world. Why don't we have such kind of a data? And he's right. So based on that, there are these 5 main pillars on which this national action plan is built upon. Now here is something, the major part of this presentation goes. Let us go to the Bhagavad Gita chapter 9 verse 7. It says, creation and destruction is the cycle that we are living in. And if we go, this cycle that I want to talk about, the traditional knowledge, the knowledge which was there in the ancient India, most of the books are not even been translated, so we do not know anything about it. At that time there was no issue of antimicrobial resistance, because at that time we might have certain kind of antibiotics, certain kind of drugs, certain kinds of Ayurvedic medicines. And when I am talking about traditional knowledge, I am not talking only about Indian knowledge. Every community throughout the world had some kind of traditional knowledge by which they were protecting themselves from the, most of the death during this era was done because of the war, because of the lack of food, because of lack of shelter. not because of antibiotic resistance. Then an era came, so it is everything, the world is going like a cyclic, this is waves, something is created, something is destroyed, something is created, something is destroyed, yeah. So, traditional knowledge, we lost it. Global epidemics, this is the era probably where there was a First World War, we don't have antibiotics, and then became an era where the first antibiotic was identified. Then we have next generation of antibiotic, next generation of antibiotic, next generation of antibiotics. So we were so happy that we have so many different antibiotics that there are no diseases which cannot be cured by antibiotic. And we were all happy. So we started abusing antibiotics. For everything we are putting an antibiotic. Even if it is a viral, put antibiotic. Yeah, so it all started over here. And then we came to an era where there was antibiotic resistance start appearing inside the patients. Then we started fearful, we are getting more fearful, and we are over here right now, we are afraid. And therefore you all are afraid. And now there are two kinds of people. One kind of people will always say कि दुनिया जैसे चलती है चलने दो. ये रास्ता आज भी ऐसा है, 10 दिन के बाद भी ऐसा रहना. Those people are ignorant people. Another kind of people are those people who will say that okay fine, there are a lot of things which are same, but there are also new things that we should also adapt. We need to know what is something latest in this world, or how can I contribute to the society, how can I contribute to research, and then their energies will dive or delve into those kind of research, and then they do something good. Options is yours. Choose wisely. So here we are afraid. We will come to a point where we are global threat, and you will see this in your life. But the objective is you should be prepared what is over here. So what kind of future I am seeing? I am seeing the future where there is an AI. I am seeing the future where there are multinational companies. I am seeing the future where a lot of money is being poured for AMR. So now, just take an example. If I get a bacteria, I give it for whole genome analysis, I will get a raw data. Analyzing the raw data will take 3 to 4 days, 5 days, 6 days, 7 days, 8 days. It can go up to a month. It can be a thesis of one student. And then you will come and say that, okay, fine, sir, I observed that there is one specific mutation in one specific gene which is causing this kind of antibiotic resistance. But now what if I'm getting the patient is going into ICU, I'm taking the sputum sample is directly going into WGS analysis. That analysis itself has its own AI. You are giving the sample, the sequencing has been done, artificial intelligence itself analyzes the data, cleans the data, assembles the reads, and it will give you the full report within 2 hours. That this person is having Klebsiella pneumonia with this number of antibiotic resistance genes. Out of these 7 antibiotic resistance genes, 4 of them are having this kind of mutation, this kind of mutation, this kind of mutation, and therefore for this kind of mutation you should have this kind of antibiotic. See, everything has to be done quickly, within 2 hours. And so that doctor can prescribe a particular medicine to that patient. और वो कब possible होगा? It will take time. Technologies take time to mature. We say that UPI came, India is great. Agree. But for UPI to be successful, computers were built 20 years back. If there were no computers, if there were no internet 20 years back, if there were no research 20 years back, then there would not have been UPI. Agreed or not? So today, we are in the era where artificial intelligence is there. The computational power is increasing. Computers are getting cheap. Microsoft is pouring in money. I will come into it later, don't worry. So we need to have that kind of future. And you are supposed to be a part of it. You are supposed to build a technology. You are supposed to use the technology in the way which is benefiting the society. And if you achieve that, then you will go to this part. And who knows, molecular medicine, there might be different antibiotics which you have not heard about. Okay, so now I will just skip all this part. I will come to this part about when I'm talking about Google and big companies. So Google, if you do not believe, Google is spending a lot of money in artificial intelligence, agreed, but it is also spending a lot of money in the drug discovery. Why Google should spend money in drug discovery? So see now, We are living in an era where a single person cannot change the entire healthcare industry. A single person cannot invent a new antibiotic. वो ज़माना गया जब अलेक्जेंडर फ्लेमिंग ने पेनिसिलिन ढूंढा. Okay, so Google is right now, it has acquired the entire AlphaFold. जो प्रोटीन आज तक क्रिस्टलाइज नहीं हुआ वो डेटा गूगल के पास है. गूगल को ये सब करने की क्या जरूरत है? Because Western world is also running behind it. और हमें ये पता होना चाहिए because you are the youth. You need to know what Google is doing in biology. It has developed a molecular syringe explicitly through artificial intelligence, where you can put your drug. That syringe, the protein is entirely AI created. You can synthesize a gene, put it into a E. coli, and now the model of a protein which was created by an AI is now being created by E. coli. You put your drug inside that vehicle, you put that vehicle into human body, it will exactly go to the location where it wants to go. That has been done by Google, even before we think it is possible. Then I hope you all heard about Zuckerberg, the founder of Facebook. The wife of Zuckerberg is running a foundation which is called as Chen Zuckerberg Initiative. They are doing a lot of research, and if you believe, there is this something which is called as CZID, and this is the data analysis done from one of the companies from Facebook. Facebook is inventing, or it is investing a lot of money into this kind of a research. So now I'm not telling you you go and work in Facebook, no. You be so skillful that you can complement these people. You can collaborate with these people because you are the future. You learn this technology and do something in India, or you collaborate, you bring part from, you give or you utilize the data of India to synthesize something, synthesize new kind of drugs for your nation. Then there is something which is called as Gates Foundation. The foundation which is run by Bill Gates. As sir mentioned, भाई CSIR और ये सब पब्लिक फंड का पैसा है, वो हमें क्यों देंगे यूज करने के लिए? वो उनकी बात भी सही है. Money is going to dry up. So we need to identify new forms of research, new forms of collaborations, new forms of linkages with whom you can work. There might be an era 10 years down the line that one of the students of IAR might say that okay fine, I am going to USA to do my dissertation with Google's AlphaFold artificial intelligence headquarters. And you need to do something about it. You need to think in that direction. I hope I'm making things more clear. So, and Gates Foundation, why I put this up? Gates Foundation, they are working a lot on tuberculosis. Why they are working on tuberculosis? If you see 4 or 5 slides at the back, which was the top organism over the Acinetobacter brumani, and then it was Mycobacterium tuberculosis. Then there is something which is called as AMR auction fund. So now this is an auction fund which is being raised by the money of different companies, where they are saying that they want to create 2 to 4 novel antibiotics by the year 2030. And which companies have invested their monies? These are the companies, big names. And the last but not the least, we have something which is also called as personalized medicine. आपको स्पेसिफिक कौन सी स्ट्रेन का इन्फेक्शन हुआ है उसके लिए स्पेसिफिक मेडिकेशन दिया जाएगा. Okay, we have technologies, but we need to mass effectiveness of this technologies is required. So we are coming over here. What is your goal? Your goal is to be prepared for this stage. Don't be fearful over here, because if you stay fearful, if you stay ignorant, then you probably not reach here. Okay, so this was the last slide, and I hope you enjoyed the session. I exactly completed at 1 o'clock, and so you should thank me.
Thank you, sir, for your rigorous and knowledge sharing experience and to complement this session. I now request Professor Buddhisagar Tawari sir to please present a memento to Goswami sir.
Thank you.
sir. With this, we complete all our expert sessions for the day. And a special mention to our esteemed speakers for concluding the sessions timely. Thank you so much, sir. As per the schedule, we will now break for lunch. I know everybody is hungry, right? Hai? So the lunch will be served at Central Lawn, where our inauguration ceremony took place, the same location. A small announcement to all the PhD and PG oral presenters. I request you to upload your presentations here in this system. We have our volunteers here, so please do it before you leave for your lunch. Yes, the PhD presentations will be held in the same room. However, the PG presentations will be conducted in the second floor of this building, which the room number is 305. So please, the PhD presenters, do upload your presentations here, and the PG ones, please upload it upstairs. I'll just brief you the schedule after our lunch. So at sharp 2, we will again meet in the given venue for our oral presentations, and then later at 4 we will have our networking tea sessions, which again will be held at the same place where our inauguration took place, and then later after that we will move to cultural event, and with that we will be concluding our today's day. So we will now move to our Central Lawn, where the sumptuous meal is waiting for you. Thank you. Have a great time.
Thank you, sir. Thank you, sir.
All the PhD presenters, please stay back and upload your presentations.
Yes, sir.